Okay, let's go ahead and get started now. We're going to wrap up the end of chapter 3 on the cells today. We're going to talk about protein synthesis. And then on Tuesday, we'll start on metabolism, which is the old chapter 24. I guess we still call it chapter 24. It's going to be about how the cells actually get their energy. Is there any way I can push back the bones test? Probably. You know, how far back do you want it pushed? I can slide it some. I think there's a lot of material to cover, yeah, I can push it back. I don't really write a note myself. Push back bone test. And we're doing pretty well on timing, so I can, yeah, I can do that. So I will, when we get out of class, I will push the bone test back. Now, as far as the testing goes, have you taken advantage of that app called the Bone Box? I sent you the link to. It's by Loyola University out of Chicago. It's an interactive quiz that you can take over and over again. It's very, very good. I've gotten some people that have taken it and used it have said great things about it. And I can't recommend it enough to you. It's free. I've sent you the link. So you really ought to take advantage of it if you can. So, okay, if nothing else, then I'm going to go ahead and I'm just glad to, so I can make that change for you. So let's go ahead and get started. Let's get into, hopefully everything will work. Not like it's been earlier in the week. Okay, now, so we've talked about everything up to the point in the cells. We talked about how the DNA is in control. We talked about proteins get manufactured in the rough ER. But what does that mean? What exactly does that all mean? Well, DNA is what we call the master blueprint, the master plan for all of our proteins. And proteins are what we're all about. Everything we do requires proteins. And so it's the proteins that make us who and what we are. Well, the order of the proteins, the assembly of the proteins is dictated by DNA. DNA consists of billions of nucleotide bases assembled in a very specific order. The order of the bases, in the DNA. We know what a nucleotide is. A nucleotide is a phosphate and a 5-carbon sugar and a nitrogenous base. There's only 4 bases. And adenine, guanine, cytosine, and thymine, and they're complementary to each other. When they pair together, when they bond together, with hydrogen bonds, they cause the DNA strand, which is a double strand, to twist. That's where we get the double helix. The order of the bases dictates the order of the amino acids in proteins. Proteins are made up of amino acids held together by peptide bonds. And they're in a very specific order. Proteins, the amino acids are in a very unique order just for that protein. A mistake in the order can have devastating consequences. I don't use that term lightly, devastating consequences. In sickle cell disease, and you'll hear more about this later on today. In sickle cell disease, we see that there is one amino acid. There's an error in the amino acid in the number 6 spot in the protein chain. Hemoglobin that carries our oxygen has 360 amino acids in its protein. Hemoglobin is a protein made up of about 360 amino acids. In the number 6 spot, on normal, healthy hemoglobin is an amino acid known as glutamic acid. In sickle cell disease hemoglobin, it is valine. A mistake of one amino acid, so putting the wrong amino acid in place of the correct one, causes the entire large hemoglobin molecule to be defective. It reduces its oxygen carrying capacity by 80%. Now, this is not something that happens randomly. This is a genetic disorder. It is a homozygous recessive disorder, meaning that the recessive gene has to come from both parents. You can't catch sickle cell disease. It doesn't work that way. But if you receive both recessive genes from your parents, and that's the only way to receive it, is to have both recessive genes, meaning your mother and your father had to be carrying the gene and pass it on to you, and you had to get both recessive genes. That's the only way a person has sickle cell disease. But what happens with that, the gene, and the gene is nothing but the physical location on the DNA strand of information on a particular trait. In this case, the trait is hemoglobin production. The gene has a mistake in it. The sequence of the bases in the gene, because that's all the DNA is. The DNA is nothing but this 6 foot long, each strand is nothing but a 6 foot long strand of nucleotides linked together with all these bases attached to other bases. A gene then is just a segment of the DNA, and the gene has an error in it. The error is that the DNA, that the sequence of the bases for the sequence of the bases codes for the wrong amino acid. And that produces sickle cell disease because it has a defect in the hemoglobin. So the order of the bases in the DNA, there are only 4 bases. The order of the bases, they can repeat over and over and over and over again, but they repeat in a very specific order. The order of the bases dictates what's going to happen in each gene. So the section of the DNA that codes for one protein is the gene. Now, genes are composed of structures known as exons and introns. Exons are the segments of the gene that actually does the coding. Introns are what we call non-coding segments that pop up in the middle of the gene. Don't know why they're there. We used to call it junk DNA. That's because we didn't know what it did. Now we know it has a purpose for something. We don't know what the purpose is yet. But we know that we actually slice out the non-coding segments. We slice out the introns before we actually start making the protein. Our cells will do this. They will automatically take out the non-coding segments of a gene before we make protein. Before we use that as the model for making protein, the instructions of making the protein. Why are they there to begin with? Don't know. We have no idea yet why we have non-coding segments in our genes. Are they placeholders? Placeholders for what? What do they do? We don't know yet. But we do know that we have the coding segments called exons and the non-coding segments called introns. The code of the gene is, again, it's specified by the sequence of the bases: adenine, guanine, thymine, and cytosine. It's in whatever order that gene codes for, but it is unique. The sequence of bases for hemoglobin is different from the sequence of bases for collagen fibers, which is, of course, a protein. It's different for the sequence of bases for keratin or, carboxylase, because its enzymes are proteins too, or, amylase or any other protein. Every protein's gene sequence of bases is going to be different and unique just to that protein. Now, the bases are organized in groups of 3. We call them triplets. So we have clusters of 3s. So we have, a cluster, like 3 guanines together, or 3 cytosines together, or adenine, thymine, and adenine, or guanine, cytosine, and guanine, or guanine, thymine, and adenine. There are 64 different possible combinations here of in these triplets. Each triplet codes for a specific amino acid. So we have all these nucleotide bases lined up in our gene. We cluster them in groups of 3 so that we have each cluster specifies, each cluster of 3, each triplet specifies an amino acid. So that simplifies the process a little bit. Instead of having a million bases here to have a million amino acids, we only have to have we have triplets. So we have 3 to a triplet, 3 bases to a triplet. So now we specify the amino acid off the triplet. Makes a little better check and balance also on which amino acid we're going to get. So the long strand of DNA made up of nucleotide bases on each side. The bases join together pair up with hydrogen bonds. They are complementary. Adenine always pairs to thymine. Guanine always pairs to cytosine. Those are their complements. The order of the bases clustered in groups of 3 dictates the order of the amino acids. Each triplet is unique for an amino acid. DNA and RNA are nucleic acids. Polymers made of segments called nucleotides. One difference between DNA and RNA is the type of Now, the reason that the double helix twists around like that is because of the hydrogen bonds occurring between the complementary bases, between the base pairs. When adenine pairs to thymine, a hydrogen bond forms there, and in the next base, maybe it's cytosine and guanine, and they pair with hydrogen and are held together with hydrogen bonds. And the hydrogen bonds are weak bonds, but they're strong enough to cause the 6 foot long molecule to twist and form the familiar double helix that we know. The sequence, though, is more than just important. It's very, very significant. The sequence of the bases dictates the order of the amino acids that are assembled to make the proteins. And that's what a gene is. A gene is nothing more than the physical location on the DNA strand with the instructions on how to make a unique protein for us. DNA is double-stranded. It uses the 4 bases, adenine, guanine, cytosine, and thymine. RNA, which does all the heavy lifting for the DNA, is a single strand. It doesn't, it does its work outside the nucleus. The DNA never leaves the nucleus. The DNA sends instructions outside the nucleus using RNA. RNA is a single strand. The only difference is that it's a 5-carbon sugar. It's a ribose sugar, meaning it has an extra oxygen. The DNA is a deoxyribose sugar, so it has lost an oxygen. It's still a 5-carbon sugar. The other difference is that there is no thymine in RNA. The RNA uses uracil in place of thymine. DNA has thymine, RNA has uracil. Those are the only differences. Now, there are 3 types of RNA. Messenger RNA, which actually carries the message to the endoplasmic reticulum. That's why we call it messenger. Ribosomal RNA, which makes the ribosome, and transfer RNA, which brings the amino acid together. So messenger RNA, single-stranded. Well, they're all single-stranded. It is a copy of a complementary copy of the DNA. The DNA opens up, the double strand breaks apart, and the genes are on either side of the strand. And the side of the strand, the gene that we want to copy is on one side. So we make a complementary copy onto the messenger RNA that's what we do. We store the information from the DNA onto the messenger RNA. We keep the sequence. We keep the triplet sequence of the bases that we had in the DNA, except that it's a complement. We call the triplet in the RNA a codon. Ribosomal RNA, also a single strand. We use it to assemble the ribosomes. And this is where messenger RNA goes to actually build have the protein built. And then the transfer RNA actually brings the amino acids together. It has a special location on it with what's called an anticodon at the bottom of its head. It has a cluster of bases called the anticodon because it is the complement to the codon on the messenger RNA. So protein synthesis then involves two parts, transcription and translation. Transcription is copying the information from the DNA to the messenger RNA. Translation is using those instructions to make the protein. The messenger RNA is the recipe. Messenger RNA is the recipe, and you have to follow the instructions on the recipe to make something. And that's what the translation is all about. You translate the written instructions to a finished product. Transcription takes place inside the nucleus. Translation takes place in the ribosomes on the rough ER in the cytoplasm. So transcription, transferring the information, transferring the sequence of bases in the DNA. Transferring the sequence of bases in the DNA onto a complementary copy onto the RNA, which means if the DNA has a base, has adenine in its sequence, what shows up on the RNA is going to be uracil. If the DNA has thymine, the RNA has adenine. If the DNA has guanine, the RNA has cytosine. When we build the messenger RNA, we are making a complementary copy of the DNA. So complementary means that we have the opposite. We have if it's adenine, we have uracil in RNA. If it's thymine, we have adenine. If it's cytosine, we have guanine. If it's guanine, we have cytosine. That's the complement. We build the messenger RNA is the complementary copy of the DNA. Now, to start this ball rolling, we have to open up the DNA strand. DNA, because it's a double strand. We have to open it up. Remember how we did it in semi-conservative replication. We open up the DNA strand, we expose the gene that we want to copy. We probably use helicase or something like that to get it to open up. And then we start building the RNA off of the DNA template. There is a promoter gene. There is a promoter sequence on the gene. I shouldn't say promoter gene, but I'll just say, there's a promoter sequence at the start of every gene that says, hey, start here. Don't start at the gene next to me, but start here. There's no guarantee that what's next to the gene is going to be correct anyway. So we have a promoter spot. Start here. We start copying, making a complementary copy of the DNA at the promoter spot. The DNA strand has a promoter. The promoter doesn't show up on the RNA. We use RNA polymerase to assemble the nucleotides. We've already made the nucleotides inside the nucleus. We've already made them. They're all floating around, ready to go. A nucleotide is a phosphate and a 5-carbon sugar and one of 4 bases. So we start assembling the complementary messenger RNA strand based off of the DNA strand. And we are following the order of the bases in the DNA. So we produce a strand of messenger RNA that has the complementary order. If the sequence of bases on DNA goes cytosine, guanine, adenine, CGA, then the sequence on the complementary sequence on the RNA is GCU, because the complement to cytosine is guanine, the complement to guanine is cytosine, and the complement to adenine in RNA is uracil. So we just continue the process. Whatever the base is on the DNA, we assemble the complement in the messenger RNA. And we use RNA polymerase to glue this together. And we assemble the strand in here. So here we see we've opened, and this is a strand of DNA. And the first step we go through is initiation. The RNA polymerase causes the strand to separate along with the helicase, opens it up. RNA polymerase allows it to separate and then starts gluing the nucleotides in place to make the messenger RNA. The next step is elongation. The first step is initiation, next step is elongation, and we build down until we reach our termination signal. We have a stop signal. And the stop signal then says, okay, that's the end of the gene. We don't need to go any further. So if you watch this, we'll let it start over here again. The DNA strand will open up, and then we will start at the promoter location, and there it opens up. We start at the promoter. We start gluing the nucleotides in place. In the red, the red stripe there is the RNA, and those nucleotides are being assembled as the complement to the original strand on DNA. We are using the gene is the segment of the DNA on the bottom, and we're building the complement off of that. The red stripe is the messenger RNA, and those bases that are being assembled there are the complement to the original bases. Once we get done making the messenger RNA, then we have to clean it up. Because remember, the DNA contains segments called introns and exons, coding segments and non-coding segments. We have to slice the non-coding segments out of the messenger RNA because we've copied everything. We want to take out the stuff that's not relevant. We don't know why the non-coding segments are there. 10 years ago, we called them junk DNA. We don't call them junk DNA anymore. We call them non-coding. We don't know why they're there. They have a purpose. 10 more years, we'll probably know what they're doing. But what we do is we've made the messenger RNA. Now we use these proteins that are called spliceosomes that slide up and down the messenger RNA and slice out the non-coding introns and glue the exons together. I just think this is fascinating how they discovered this too. So we slice out the non-coding introns and glue the exons together. And that wasn't a Friday afternoon name, spliceosome. Because what are they doing? They're splicing the segments together. And instead of giving some sophisticated, elegant name for its action, spliceosome. It's a slicer and dicer. It's like, okay, if we name this thing, we can go home now. So let's call it spliceosome. I also think they probably named that so people could remember what it does. So anyway, we slice out the non-coding segments. We put together the coding segments, and now we have a processed messenger RNA strand that is ready to leave the nucleus and go to the ribosome. When the messenger RNA goes to the ribosome, we call it translation. We're taking the instructions from the DNA out on how to make a protein to the ribosome. We're going to assemble our protein there. We're not allowed to take the DNA out. We can only make the copy and take that out. Sort of like if you want to get a recipe from grandma. Grandma has this recipe for these great biscuits. And so, but she won't let her cookbook out of the house. It's not that she doesn't trust anybody, but she doesn't trust anybody. So what she'll do though is she'll either give you a copy of the recipe or let you make a copy of it yourself at her house. Maybe she's got a printer with a scanner because she's technical, just because she's grandma doesn't mean she isn't tech savvy. Or she makes you write it down, or she writes it down for you. Either way, you have a copy of her instructions on how to make biscuits. Now, maybe her original instructions were based on using a gas stove. And gas, if you've ever used a gas stove versus a electric stove, you know that there are differences. And so maybe her instructions, she copies everything down and then she crosses out the instructions for how to bake the biscuits in a gas oven as opposed to an electric oven. Or maybe the recipe is her grandma's and the recipe is over 100 years old, and the instructions talk about using a wood stove. Because it goes, maybe the recipe goes way back, and so she crosses out the instructions on using a wood stove. Who knows? Whatever it is, she removes the stuff that's not relevant, and what's left over are the relevant instructions that you need. Maybe she doesn't like you, or maybe she just doesn't want her recipe to get out. So she leaves out a few other instructions too, but that's a whole other story. So anyway, to make our protein, we now have the instructions on the messenger RNA. We have the sequence of bases on the messenger RNA, which represents the order of the amino acids that need to be assembled. So whatever amino acids we need, they're already there, they're already available, and we will assemble everything in the correct order. Now, it is true that if the amino acid isn't there, we will not make the protein. We won't make a part of a protein and then stop. If we don't have all 20 amino acids that we need, we're not going to make the protein. Anyway, so we have the messenger RNA, we have the amino acids, we have the transfer RNA, and we have the rough ER, because that's where the ribosomes are going to be. Now, the code on the messenger RNA is very important. The triplet of the bases on the DNA is represented by the codon on the messenger RNA. It is a complementary sequence. Remember, if the DNA has, GGG, guanine, 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 its complement that shows up on the messenger RNA is going to be cytosine, 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 C, C, C. On the RNA, we call that triplet the codon. So we have the triplet on the DNA and the codon on the RNA. Now there are 64 possible codons, 64 possible codes for amino acids. Now there are only 20 amino acids, 64 possible combinations for 20 amino acids. Now, and so how do we get 64? Well, there are 4 bases and they're organized in clusters of 3. And if you take 4 to the 4 bases to the 3rd power, you get 64. Okay, so just take it at face value. We have 64 combinations, 64 possible codons. 3 of the codons are stop codons. They are termination codons. So we really only have 61. But the rest are, the 61 codons are specific for the amino acids. Now, there are only 20 amino acids. So what that means is we have a lot of codons that code for the same amino acid. We have a lot of redundancy. As we see on the chart from the next slide, you're going to see that there are some amino acids that have 6 different codons that code for it. Some have 4, some have 6, some only have 2. One only has 1 codon. Why do we have this? Well, in case there is a mistake in the transcription, for some of the amino acids, even a little mistake of putting the wrong base in the sequence may be harmless because it still may code for the same amino acid. So you'll see here when I change the screen that, as I said, some of the amino acids have up to 6 different codons. Now here, this is the code right here, the triplet code. Each codon, which consists of 3 nucleotide bases, is for a specific amino acid. Now this, before you look at this and say this is too confusing, and it is at first glance. But if you look at it, you say on the left-hand side, it says first letter, and it has guanine, adenine, cytosine, and uracil going up the left-hand side. And the second letter says uracil, cytosine, adenine, and guanine going across the top. And then down the right-hand side, you have all 4 of the combinations there. So let's say if you have, I'll just make up one. Let's say we have UAC as a possible codon. So the first letter of UAC is U. So that takes us to this row right here. The second letter is A, which takes us to this column. UA, and the third letter was C. So we go over here and we find C, and C lines up here. UAC, uracil, adenine, and uracil, codes for the amino acid tyrosine. So does UAU, codes for tyrosine. So in this case, we have 2 different codons for the same amino acid. How about GAC? Okay, so the first letter is G. So it's in this this row. A is this column, and C is the second letter down. GAC codes for aspartic acid. So does GAU. How about CGU? Pick another one. First letter is C. Second letter is G. Okay, CGU. Third letter is U. So there's C. There's C. That row. G, U. Arginine, right there. CGU is arginine. So is CGC, CGA, CGG, as well as AGA and AGG. So arginine shows up a lot in our protein, in our proteins, because there's 6 different possible combinations that make arginine. Some only have 1. Methionine only has 1. Methionine is in every protein. It is the initiation codon that starts the process. We see lots of these have multiple codons. Leucine, the amino acid leucine, has 6 possible combinations. So does arginine. Valine has 4 combinations. Isoleucine isoleucine has 3. Let's see. Glutamic acid has 2. Glycine has 4. Arginine has 6. Cysteine has 2. Histidine has 2. Glutamine has 2. So nearly all of these have at least 2 different combinations of codons. Depending on the amino acids we probably use more frequently, like proline has 4, serine has 4, threonine has 4, alanine has 4, glycine has 4. All of these, the ones that we use a lot have many combinations. The ones that we use infrequently have only a few combinations. But every amino acid has a has at least one unique codon combination. So the messenger RNA, the sequence of the bases, are going to form codons, clusters of 3, and the sequence of the bases form the unique codons for the specific amino acids. So this is the code. This is all 64 combinations. Here are the 3 stop codons, UAA, UAG, and UGA. All 3 of those stop, mark any one of those present will mark the end of the messenger RNA strand. Now here is transfer RNA. Transfer RNA has this unique shape. It's been described as a fishhook or the outline of a guitar or whatever. What's important is that there are 2 ends on it. The tail end is where the amino acid is located. So the amino acid is located up here at the tail. At the head end, at the bottom of the strand, and these are all nucleotide bases here that make up the transfer RNA. But at the bottom, at the head end, this is the head, there is a sequence of 3 bases that form the anticodon. The anticodon is the complement to the codon on the messenger RNA. So if the codon is AUA, if the codon is AUA, then the anticodon is UAU. So because the complement to uracil is adenine, the complement to adenine is uracil, and the complement to the uracil is adenine. So AUA is the complement to the UAU. So the anticodon is bringing the transfer RNA with that unique anticodon is bringing in the amino acid. And the anticodon is the complement of the codon. So the anticodon is the complement of the codon. The anticodon on the transfer RNA is bringing in the amino acid. The anticodon on the transfer RNA will bind to the codon on the messenger RNA inside the ribosome. Again, if the codon is AUA, the anticodon is UAU. That's the only combination that can bind there. AUA specifies the amino acid. AUA specifies the amino acid. The anticodon will bring that amino acid to that codon that it's complementary to. So if your codon is AUA, the only thing that will bind an amino acid there is an anticodon with UAU. So the ribosomes then allow the messenger RNA to create in other words, the ribosome tends to slide over top to surround the messenger RNA and slide over top of the messenger RNA as it exposes a binding site for the amino acids and the complementary codons. Okay, so what's that mean? What did I just say? Messenger RNA has to bind with the transfer RNA. The codons on the messenger RNA will bind briefly with hydrogen bonds to their complement found on the transfer RNA. So for example, so if AUA is the messenger RNA codon, its complement is UAU on the transfer RNA. They will bind together briefly with hydrogen bonds. That will allow an amino acid to be brought to the transfer to the ribosome. When the second transfer RNA comes down and lands on the messenger RNA, it will also have an amino acid. And those two amino acids will link together with a peptide bond, and then a third amino acid will come down on a third transfer RNA. And now you'll link the first two amino acids to the third one, and this process will continue until we hit the stop code. Now we have initiation, elongation, and termination as our sequence, just like we had in transcription. In initiation, we start at the promoter, at the start codon. We have the promoter spot here is called the start codon. And unlike the promoter in the DNA, not showing up on the RNA, here we have our start codon, which always codes for the amino acid methionine. Every protein starts with methionine. The anticodon, the codon of the start codon is always going to be AUG, adenine uracil guanine. The anticodon on the transfer RNA will be uracil adenine guanine. You can see here, uracil is the complement to adenine, adenine is the complement to uracil, and cytosine is the complement to guanine. So the transfer RNA that has that anticodon sequence that is a complement to the messenger RNA start codon carries the amino acid methionine, brings it down here, and it binds to the messenger RNA with hydrogen bonds. And here it has drug along methionine. Amino acid number 1 in our protein. We have a second binding site exposed right here next to it. Here is the codon for the second binding site. The codon is GUA. The anticodon is going to have to be CAU. Because remember, guanine code is complement to cytosine, uracil is complement to adenine, and adenine is complement to uracil. So whatever lands here is going to have the anticodon of cytosine, adenine, uracil. Here's another animation that shows us. There is our ribosome. The ribosome forms with a small subunit. And here comes the methionine landing at the start codon right there. The second codon in this sequence is going to be CGC. So the anticodon is GCG. As the second transfer RNA lands, the two amino acids link together with a peptide bond. When a third one comes down, we link the first two to the third one. Let me show this again. There's our messenger RNA. Here comes the first half of the ribosome called the small subunit. Here comes the methionine landing on the start codon. Here comes the large subunit of the ribosome. Looks like a hamburger bun. We have a binding site available here. Arginine is attached to this transfer RNA with a binding site down here. The ribosome slides across the messenger RNA and keeps exposing the second binding site there. And the transfer RNAs will keep coming down. And what will happen is as the transfer RNA's anticodon binds with the messenger RNA's codon, the transfer RNA will pause there long enough for the amino acids to start linking together. See, here comes the first amino acid, methionine. It will link to the second amino acid, arginine, with a peptide bond. Then methionine is kicked out. Methionine and arginine will link to the third amino acid that's presented there, whatever it is. on the anticodon CCU, here comes lysine. They will link to glycine and will kick out the second transfer RNA. The third transfer RNA will be kicked out too when the fourth one comes in. And it all depends on the sequence of the codons. The messenger RNA has this unique sequence of codons, and we have the complement to those codons called the anticodons on the transfer RNA. That tells the transfer RNA what amino acid to bring down to the codon. And we start the sequence at, we have the start codon, and that's where we start. We always start with methionine. Now, elongation, we continue to build the protein. Whatever the messenger RNA will slide through the ribosome, or the ribosome slides across the messenger RNA. It doesn't really matter. But every time every time a transfer RNA lands on a binding site, the ribosome will move, exposing a new binding site, like there where the letter A is. That's the new binding site for the next available transfer RNA to come down. Go back one and do that one over again. So let's hit it. So you see how we slide over and expose the A site again. And every time we're landing here, every time we have a peptide bond forming between the amino acids up here, we kick out the old transfer RNA and expose the site for the new one to come in. Our stop codon, we had a start codon, now we have a stop codon. The stop codon uses one of the three stop codons from the list, UGA, UAA, or UAG. An easy way to remember that is if you're a Georgia fan or not, think of UGA, think of Georgia, think of UGA, their bulldog. Doesn't matter. The UAAs, the UAGs, and the UGAs are the stop codons. When we hit the stop codon, the process of building the protein stops. Back here again, because when we bring it up here in this next example, there'll be a stop codon right here. UGA is the stop codon. We hit that, and that causes us to release the protein from the ribosome, causes the ribosome to separate, and will eventually, the messenger RNA will break apart into its respective nucleotides to be reused. And so we end up stopping the action. We've made our protein. We had a promoter on the DNA strand. We have a start codon on the messenger RNA strand. We have a stop codon on the messenger RNA strand. And the transfer RNAs, which contain anticodon heads that are complementary to the messenger RNA, are bringing in the amino acids. And this takes place in the rough ER. And then when the proteins are assembled, they are gathered in a transport vesicle, which then is delivered to the Golgi apparatus. Now, so what we do is we take the DNA gene, the gene in the DNA that contains the instructions we're looking for. We want to make hemoglobin, we want to make oxidase, we want to make keratin, we want to make collagen, we want to make something, an enzyme, whatever. So we're doing normal maintenance and repair in our bodies. So we're making this protein. We go to the gene, it has its information. We have multiple locations of these genes throughout our DNA. We don't just depend on one spot. The gene is nothing but the physical location of the instructions, the physical location of the sequence of the bases in the DNA. We open up the DNA. We use RNA polymerase to assemble the RNA complementary copy. Whatever the base is in the DNA, the RNA has the complement. We take that messenger RNA, after we splice out the non-coding segments, we take that to the ribosome. So we have the codons on the messenger RNA, which are the complement to the triplets on the DNA. If CCC was the triplet, the codon on the RNA is GGG. It is always the complement. But we bring in the transfer RNA. The transfer RNA contains the anticodon on its head. The anticodon is complementary to the codon on the messenger RNA. And here's where we get back to the original sequence in the gene in the DNA. The anticodon base sequence is identical to the sequence of bases in the DNA. We've come full circle. We take the base sequence on the DNA strand at the gene, we complementary copy it to the RNA, to the messenger RNA. Again, the complementary copy means if the DNA has CCC, cytosine, 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 as its sequence for one triplet, the complement to that on the RNA will be guanine, 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 or GGG. But so the codon on the messenger RNA would be guanine, 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 or GGG. The anticodon on the transfer RNA will be the complement to the messenger RNA sequence. And the complement to GGG is CCC. So we bring us back to the original sequence after all on the transfer RNA. The only thing different is we use uracil instead of thymine, but it is still the sequence. So we don't change anything. You know, we're still following the sequence specified by the DNA to build the protein. And it works. Now, we have redundancy. We have multiple gene locations, so we're not tied to one spot only in case something happens to that gene at one location. We have redundancy in the sequence of the bases for the various amino acids. You know, some amino acids have 6 different combinations. Many have 4 different combinations. So even if there is a mistake in the base sequence, in many cases, it won't matter. It'll still code for the same amino acid. And if it doesn't code for the correct amino acid, then usually the protein doesn't get made. It shuts down at that point. If there's a mistake in the coding, you know, if a mistake gets made in the coding, we'll just stop. We won't go any further. Now, if a mistake occurs in the DNA, then we have a more significant issue. We call a mistake in the DNA is called a mutation. Mutations happen. We talked about them the other day, you know, from radiation, from smoking, from pesticides, from pollutants, from viruses. All these can cause mutations in the DNA. A mistake in the RNA may only happen one time, and it may happen in an area that is redundant, so that even if you have a mistake in the base sequence, perhaps it still codes for the same. It still codes for the same base, same amino acid. Here we have a strand of DNA with cytosine, guanine, adenine, adenine, cytosine, 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 guanine, and adenine. That's okay. That's fine. What happens if we substitute in a different base? Here we have, the substitution occurs in the gene sequence right here. Cytosine, guanine, adenine. A second cytosine goes in place of the guanine. And what does that do? Well, what it does is it causes a change in the sequence by coding for a different amino acid. CGA will code for arginine, and CCA codes for proline. Now we have a significant difference here in amino acids. What does that do to the protein? Well, it may completely destroy the protein. And it may, what it means is if we have a substitution here, this is a substitution mutation. The gene is effectively destroyed. If we put the wrong base in the gene, take out the good base and put in a new one that's wrong, we end up with a gene that's defective. So, and unless we can cut that out, and we do have the ability to move up and down the DNA and cut out mistakes and repair them. But if we don't get it repaired, then we have this damaged gene. Now, did that damage affect only that gene, or did it affect all the genes, for that particular trait in our bodies? So we don't like substitutions. We don't like putting, because a substitution affects the gene. It doesn't affect the messenger RNA. It affects the gene. So here we see what happened with the substitution with hemoglobin. This is a substitution mutation, the situation with hemoglobin. Hemoglobin has, in its number 6 spot, this is the sequence of the amino acids in the protein hemoglobin. The number 6 hemoglobin is glutamic acid. Number 6 spot in hemoglobin is glutamic acid. Number 6 spot in sickle cell disease hemoglobin is, or what we call hemoglobin S, is valine. Now, how did that happen? Well, it was a very simple substitution. Adenine replaces thymine in the defective hemoglobin. Thymine, here's normal hemoglobin here with thymine, cytosine, thymine, cytosine. In the hemoglobin S, we have adenine put in place of thymine. And what this does is that causes the gene to code for valine and not glutamic acid. So we have our defective hemoglobin that is reduced its oxygen carrying capacity by 80%, has become brittle and twisted and curved and shaped like a sickle or a crescent shape, and we end up with our crescent shape or sickle shaped red cells. So one little mistake, one base difference, but we substituted thymine in for adenine, and it makes all the difference in the world here. Now, this is only going to occur when it's as an inherited disorder. It is not something that is acquired. It only occurs when it's inherited. You have to have both recessive, it's caused by a recessive gene. You have to have both recessive genes from your parents to have this condition. It's the only way. And it's such a little mistake. It's such a very little mistake here. It's like, and it really doesn't matter if it's the number 6 amino acid or the number 60 or 300th amino acid. The mistake would occur regardless. The defective hemoglobin will be made regardless, but it is, it happens early on. It doesn't make any difference. It just happens so early in the production of the protein, and yet it ruins the whole protein. The other type of mutation that we encounter are the frame shift mutations. Frame shift mutations occur when we either cut out or insert a set of bases. We can insert bases, because we can insert nucleotides, or we can remove nucleotides. Here is our sequence up here. GCT, AGT, ATA, CGG, and CGA. That's our sequence. Down here's the complement to the sequence on the DNA. So what happens when we add a set of nucleotides? We put them in right here after the third triplet. We've added guanine and cytosine. Cytosine is the complement of guanine. So we've added guanine here. That pushes everything down. That's the frame shift. We shift everything down. So now instead of ATA, the third triplet is GAT. The fourth triplet isn't GGG, it is ACG, because now it includes these three. The fifth triplet isn't CGA, it's GCG, because we've slid everything down one. So now our order is totally destroyed. The order is totally destroyed, and now we're going to be coding for totally different amino acids in this protein. This destroys the gene. The gene can no longer be used when you have a frame shift mutation, when we've inserted an extra set of nucleotides, or if we've cut out an extra set of nucleotides. We shift. If we cut out a set, we shift down again. Imagine if we cut out A and T at the beginning of, cut out the A and T, and cut out the first A and the first T on the third triplet. ATA slides down. If we cut out the first A of ATA, we slide down one, and now our triplet is not ATA, but TAC. And then triplet after that is GGC instead of CGG. So again, whether we cut out the triplet, cut out the nucleotides, or add in the nucleotides, we have altered the sequence below the frame shift, and we are no longer have an effective protein. If the protein does assemble, it will be defective, it will be junk protein. Usually what happens is the gene stops, the sequence stops, and the gene is essentially destroyed at this point. It's still there, but it's no longer functional. So this is the frame shift mutation. We know that cells can undergo two terms. Differentiation, that's when they grow into, heart, lung, muscle, skin, and so on and so forth. Determination is when they have not started growing, but they have already been genetically predetermined as to what they're going to do. For example, when we are fertilized, the egg and the sperm combine, and we are one cell. We are a fertilized egg now. And that's in the beginning of the fallopian tube of our mothers. By the time that fertilized egg reaches the uterus, 4 inches away, it takes 7 days to go 4 inches. It's no longer one cell, it's 160 cells. It has an outer layer, an inner layer, and a fluid center. And it implants itself on the wall of the uterus. It has already started growing into structures. Many of the cells have already are starting to become differentiated into structures. Other areas are already predetermined as to what they're going to become: bone, lung, skin, muscle, whatever. And the cell, we are also a mass. We are a very unique, specified, differentiated mass of 160 cells. That's at the end of the first week. By the end of the third week after fertilization, we're about, we're like 3 or 4 times bigger, but we have a heart that's pumping blood. We start seeing the beginnings of our hands and feet and arms and legs and head and so on. By 6 weeks after fertilization, we have a complete skeleton in place, we're about a ½ an inch long, and we have our very distinctive shape of looking like a little tiny human, which is what we are. We are from the point of conception. But at 6 weeks, we definitely have everything in place, and then it just has to grow. So differentiation and determination. Differentiation is growing into structures. The determination is the code is there. It just hasn't gotten turned on yet till we implant. Cells grow when needed. It's called sometimes we have excessive cell growth called hyperplasia. Sometimes they don't grow very fast at all and they shrink. Hyperplasia, we grow cells rapidly when we need like lots of red cells. For when we're exercising, at a higher altitude, or lots of white cells that we're fighting an infection, or atrophy where the cell shrinks in size with disuse, like muscles shrink if they're not used, because the muscle cells will shrink in size. And the last thing talks about aging of the cells. There's a variety of theories about this: wear and tear, mitochondria being damaged by free radicals, autoimmune disorders. But my personal favorite is the telomeres. Telomeres, those caps on the ends of our chromosomes, generally wear away as cell goes through, mitosis and cell division. Every cycle takes away a little more takes a few more telomeres off at the end of the DNA as it gets replicated, and eventually the cell stops growing, and the cell line stops when the telomeres shorten enough. Now, cancer cells don't play by the rules. Cancer cells have an enzyme called polymerase, which keeps rebuilding, because rebuilding their telomeres, because cancer cells grow at a rapid rate and reproduce and reproduce and reproduce. And you would think that they would burn themselves out because they're going to destroy their telomeres. Well, they keep rebuilding their telomeres, so they can continue to grow and grow and grow. We normal healthy cells don't have that advantage, and so our cells gradually wear out, and that is the end of our chapter on cells. So let me get out of here. Anyone have any questions on anything? Yeah, I'm going to go move the bone test back as soon as we get out of class here. Okay, then I'm going to get us out of here. Lab this afternoon is going to be on the joints and articulations of our bones, where they rub together and how we make how our joints work. So see you in lab then.